thanks to the organizers for letting me have this opportunity to talk today. I have noted that in the program I have relocated to northern part of Norway. I don't think my family knows about it. I was actually removed from the hospital where I worked in because they didn't want me to treat the patients that I was treating. But relocating to Norway, I wouldn't mind. It's a nice place, but so it is. Anyway, before I got into this, I led a quiet life looking at patients with autonomic dysfunction. And then a woman like this came along. She was 18 years old at the time. She was a runner-up for the national soccer team. She developed burning pain and muscle weakness within 7 days after HPV vaccination. Subsequently, she had progressive muscular weakness, chronic severe headache, orthostatic intolerance, fatigue, exercise intolerance, cognitive dysfunction, widespread involuntary muscle activity, and severe pain. She had to quit her studies at the university. She lost a bit of weight. She was then dependent on help 24/7. She was diagnosed in my clinic with POTS and with ME, and she participated in a Danish TV documentary called The Vaccinated Girls, and that caused a lot of commotion and sort of forced the health authorities to put up centers for these kind of patients. So in June 2015, we had regional centers established, one for each of the 5 regions. I was in the capital region of Copenhagen, and I was appointed to be a leader of that center. We had seen some patients from 2011 and onwards, and the irony was that at that point in time, I was doing studies for Merck on HPV vaccination. I was involved in 8 studies and vaccinated about 3,000 girls, so I got a bit of bad conscience by that. I've seen so far 845 patients with possible side effects. The center was closed in September last year, and I was sort of thrown out, and now I'm at my private clinic, and I'm doing research at the National Hospital. These are the ages of the patients that we saw. And as you can see, most of our patients were from the catch-up group. That would be 16 to 26 years of age. Not that many in the target group between 10 and 14, but that's the way it looks. These are the main symptoms. I've seen that so far I've analyzed 150 patient records. I will analyze all of them. But as you can see, there's a number of symptoms: fatigue, headache, cognitive dysfunction, orthostatic intolerance, nausea, sleep disturbances, palpitations, abdominal pain, reduced muscle strength, bladder dysfunction, and the list goes on. I've only taken those that are over 60%, affecting over 60% of the patients. And we have so far looked at how many of these will fulfill the criteria, the IOM criteria for ME/CFS, and that's about 80%. So I think we have a group here that have developed ME following HPV vaccination. Maybe the vaccine has been the trigger. We don't know, but that's the way it is. So one of the younger doctors in my department, she said, well, why don't we do this in a systematic way, which is a very good idea. So we started looking at their autonomic symptoms using a questionnaire called COMPASS 31. It's been developed by the Mayo Clinic, and it's a very solid, consistent questionnaire for autonomic symptoms. And when we look at our patients over here, you can see compared to the control group of vaccinated girls, young women without symptoms, there's a clear distinction between the groups. Our patients have more symptoms than patients with diabetic neuropathy, who are supposed to have autonomic disturbances. They have more symptoms than systemic sclerosis and multiple system atrophy, which is a disease that severely affects the autonomic nervous system. So there's no question that these patients have severe autonomic symptoms. The other questionnaire we started a little later to use was the fatigue scale for motor and cognitive function. It's a scale that has been developed for multiple sclerosis, but it has shown a consistency and has been translated into Danish, so that's one of the reasons we use this. And if you look at that and compare our patients over here, which I call HPV/ME, compare that to multiple sclerosis, they have the same degree of fatigue. They have more fatigue than post-stroke patients. And you may know that in post-stroke patients, one of the major problems is fatigue. So these patients of mine are really sick. And you can, if you look at compared to the vaccinated control, there's no question that there's something wrong. And if you look at the severity of the fatigue, you can put that into 4 categories: no fatigue, mild, moderate, and severe. And most of my patients fall in the severe category. So these are really severely affected patients. So now we've sort of established that these patients are ill. They have autonomic symptoms. They have severe fatigue. Now we wanted to find out what's wrong. So we had to go back. I studied immunology in 1975. Lots of things had happened since, so I had to reread the whole thing. And one of the things that we started with this cartoon showing up here that this is what happens when you vaccinate. You give the antigen together with the adjuvant, and that activates these antigen producing cells, the cells that are called dendritic cells. You've heard about those. They present the antigen to the T cells via the tissue types or HLA types. And so we focused on that to begin with. And we looked at tissue typing in 180 patients, compared that to almost 300 controls. And we found a difference in a specific HLA type, which a Norwegian group has also found in ME. So there's a comparison there. And what does this tissue type signify? We know it's involved in type 1 diabetes. We know it's involved in celiac disease. And somewhat ironically, it's also involved in cervical cancer. So we think from this that we have, from the first step, found a tissue type that's associated with autoimmune disease. So the next point would be to see, are there any antibodies out there? Because what we propose is that after they have been presented with the antigen, it moves forward to produce antibodies. So the first thing we looked at was a non-specific antibody called antinuclear antibody. And as you can see, compared to a control group of blood donors, these 83 HPV patients were positive. 60% of them were positive for this antibody. And especially in one fraction of the antibodies, it was very positive compared to the background population. And if you look at what's normal, this is the age group that we have. Normally, you won't find these antibodies in patients under the age of 34. Even when you get older, you have increased antibodies, but it's nowhere near what we found in our HPV cohort. So the next thing was to look at, can we find specific? We know that this antibody is involved in a number of different diseases, autoimmune diseases. Now we wanted to look at what kind of antibodies would these be. So we found a non-specific signal, and the next is to look at the specifics. And let's see, nothing is happening. There we go. That's at least the first part of it. There's a saying that to a hammer, everything is a nail. And as I am interested in cardiovascular control and autonomic dysfunction, I looked at what happens to blood pressure and heart rate. And this is a simple stand test where you stand up, blood pressure falls, heart rate goes up, heart rate comes down again, but at a somewhat higher level. And if you look at an HPV patient, you don't have to be a physiologist to see that there's a large difference. What you can see is the blood pressure falls, heart rate goes up, heart rate tries to come down, and then it starts to oscillate. And the same happens with the blood pressure. And if you do that in normal subjects, you get mentally fogged. You lose the normal association between activity and flow in the brain. It's been shown by a group in the US. So there's some of the explanation here. So of course we were interested in cardiovascular diseases and looked at what kind of antibodies do we know are involved in cardiovascular disease. So we found these G protein-coupled receptor antibodies that are involved in dilated cardiomyopathy, in hypertension, and in preeclampsia. So those were the ones we were looking for. And we found this very clever old gentleman in Berlin who helped us out. And the way he did that is he uses a bioassay. He takes out cardiomyocytes from newborn rats. And then if you treat them nicely, they will go on beating at a rate of 140 to 150 per minute. And then you can add different substances to it and see how it reacts. So this is an example. You have the basal level beating at 148 beats per minute. You add the IgG, the autoimmune fraction, as it were, to this preparation, and the heart rate goes up to 232. Then you add a beta blocker. So you block the beta receptor in the sympathetic nervous system, and you see that the heart rate goes down. So in that way, by adding blockers to the system, we can find out what kind of antibodies are in there. It's a complicated process. It takes about 5 days for each sample. What we found was we found antibodies against the beta-2 receptor in the sympathetic nervous system, as has also been found in patients with ME. We found M2 receptor antibodies. There have been M receptor antibodies found in ME. And we are the first who found an antibody against the nociceptin opioid peptide receptor. It's a receptor that's involved in the pain, and maybe it's a candidate for the fibromyalgia. We can talk about that later. But if you look at the group of patients here who had at least one of these antibodies, it's 96%, and it's about 28% in those girls that were vaccinated but did not have symptoms. What happens when you stimulate such a receptor? Well, if you stimulate the receptor by beta stimulation and without antibodies, you see an increase in the energy production called cyclic AMP, 25-fold. But if you add the IgG from the patient, you see an increase of 75-fold. So there's a lot of pressure on the cells to produce energy all the time. They keep the response longer, and it has a higher response in all these cells that are innervated by the autonomic nervous system. This is a bit slow because it's a big slide. I think that's why. It takes a little while before it comes up. Okay, so this is an experiment that we didn't plan. We were asked by a lung doctor to look at one of his patients who had asthma. And he said that every time she takes her asthma spray, she faints. So could you please do a tilt test and see what happens? So we put her up on the tilt test. This is the blood pressure, heart rate. We tilt her up here. Everything is okay. She takes her first dose of salbutamol, which is a beta-2 receptor stimulant. And as you might note, she starts to oscillate in her blood pressure and in her heart rate. And then, as is usual in asthma, you take your second dose. And when she took that, she vasodilated, lost her blood pressure, and was unconscious for half an hour. So there's sort of a biological way to test. We're not going to do that anymore, but that's a way to test whether you have these antibodies in the body. So we found a difference in the tissue type associated with autoimmune disease. We found a non-specific signal of autoimmunity, and we think we found specific antibodies directed against the autonomic and somatic sensory system. And why do we have this? Well, there are several hypotheses out there. One is the molecular mimicry, because there's an overlap between the protein contents of the HPV vaccination and the human proteome, so there might be cross-reactions. The other thing is that you can have what we call bystander activation, and we know that the HPV vaccine is very powerful in increasing the cytokines. When you vaccinate, you increase them 10,000-fold as compared to what you would do in a natural infection, and that might cause other cells to wake up And that's what's called bystander activation. One other thing with the vaccination is that we know, or there's a theory out there that how you create an autoimmune disease is by you activate some of the cells. At some point here, you get subclinical disease with autoreactive T cells, and then you reactivate them again at a later stage, and then you get the aggressive T cells, and you get the autoimmune production. So that's what happens when you give HPV vaccination. You vaccinate, and then you repeat it once or twice. And most of our patients have got their disease after the second or third vaccination. So there's a theoretical background for this. So in conclusion, we have a small percentage. It's about 1 per 1,000 of HPV vaccinated girls, young women that have serious adverse events that are not really recognized. The clinical picture bears a close resemblance to ME/CFS. Our studies point to an autoimmune cause for the disease. The immunity is primarily directed against the autonomic nervous system. And I think in my clinic, it's an important finding because I can use the knowledge of these antibodies to direct my treatment and give them the sympathetic, it is sympathetic, yes, but the symptomatic treatment that they really need. So with that, I would like to thank my collaborators, Louise, who was the one who started all these questionnaires. Heike is the one taking care of mitochondrial analysis. Kim is the one who's the immunologist and taking care of some of that. And Gerd is the one who's doing all the testing with the antibodies. And with that, I would thank you for your attention.
So thank you very much. It was very interesting. So now, what about future treatment indications? And I'm looking. Let's for 5 years or 10 years from now. So could there be a possibility for immunoabsorption? And can you first explain what it is and if there is a possibility?
I think we have a smarter way of getting rid of the antibodies. We're going to test that hopefully within this year. We're going to do some preliminary studies on that, proof of concept, because what I want to know is, are these antibodies the cause of the disease or are they just there? So by removing the antibodies, we'll know more about it. And there is a component that I can get from Germany, and I just have to apply for the Danish health authorities to get the permission, and we're going to do that. Because immunoabsorption, it's a way to take away the antibodies. It's very expensive. It's difficult in the public system in Denmark. You can't get it. You can also do plasmapheresis, which is also quite expensive. You can do IV IgG, so where you infuse IgG to displace the antibodies. I haven't found it very useful.
Yeah. All right. Other questions?
You're a political dynamite.
Oh, yeah,
I think this needs to be widely known, the stuff you've put up for us, the information you've given us, because there are a lot of young children and young people being put at risk, both boys and girls, because of the demand that this vaccine be given to them without any consideration of these factors. So please, can you tell us when you're going to publish it? And please will you publish it as widely as possible?
Yes, the idea is I want to publish within the next month. I've just delivered the report to the Danish health authorities because they were the one who funded actually this research. There's not much funding in this, and as you probably know, I'm up against two things. ME is not considered a somatic disease, or was not considered a somatic disease in Denmark up until the referendum we had in March. And you don't talk any bad things about vaccination without getting a lot of things thrown at you. So I've been up against serious forces, and it has been difficult to get through. But I will publish, definitely.
Please.
My question was a bit similar. I'm sorry to hear that your science was closed down. It seemed that you had a very good lead in your clinic with this vaccination trial. But it's very difficult to do science in Denmark. It's especially difficult to do science in things that is a bit like ME/CFS because of the problematic attitude, the psychiatric attitude that is in Denmark.
Yes, well, we hope that that's going to change. I'm working together with the Danish ME Foundation, and we're trying to push the politicians to do the right thing. They've done it so far. Now we need the health authorities to accept what the politician says. It's not going to be an easy task, but we're going to work on it. But we have some psychiatrists that are really going against us because that's his bread and butter, more or less, and he doesn't want us to change that.
I know about this one. But I have one more question. You said you have some treatment to your young patients. Is there anything you can tell the audience here that is interesting for us all?
I do treat their symptoms. So if they have POTS, I treat that because that's what I know something about. I can get them to sleep using antihistamines. I can use beta blockers, which is obvious as they have antibodies against the beta receptor. It doesn't work in everyone, but it does work in, the beta blockers works in about 60% of the patients. We use pain killers. So what we're trying to do is to get their symptoms to go away. And then I have a physiotherapist who is very good at, doing pacing, which is a very slow way to get back into life. So we try to rehabilitate them. So that's sort of the arrangement I have in my clinic.
Thank you, doctor, for your service. You're very good.
Right.
Do you know what ingredient it is in the vaccine that's causing this, or you're just saying the vaccine in general?
Well, the obvious thing is the antigen, because you have the whole surface of the HPV virus, and that's very antigenetic. And you have this overlap of heptapeptides in the coating of the HPV vaccine that is also found in humans. So that's why you have the possibility of cross-reaction, giving what we call molecular mimicry. So we're looking into that. There might be other components in there. We know there's a lot of other stuff in there. And the problem is that in most of the studies where you look at HPV vaccination, you don't compare to placebo. You compare to something that is devoid of the antigen, but you have all the other stuff in there. So we don't know whether that's some of the things that causes the effect. And we're not going to get them to do real studies where they compare to saline. So that's going to be difficult. But I'm not focusing so much on that. I'm focusing on treating my patients and get them accepted by the system. And that's the important thing for me in this context.
The antibodies against the beta-2 receptors, do you think that, sorry, I'm here. Do you think they could be the body's natural response to a hyperactive sympathetic nervous system, which is constantly stimulating those receptors?
As we know, we don't know what's the hen and the egg here. That's why I want to remove the antibodies and see what happens. But I can, appreciate from what we see when we stimulate the cells with the IgG from the patients that you get this very high response. And I think that one of the things we've been looking at is whether that harms the mitochondria, because you would eventually create a lot of reactive oxygen species in there. And we try to prevent that by giving the patient C vitamin, selenium, which is two of the good antioxidants. But I think that many of the things can be explained by what we have found. But again, to a hammer, everything is a nail. I found these antibodies, so I want to explain everything by the antibodies. Others would explain everything with the TRP3, and so there are different ways. We don't know. Maybe these antibodies would cause changes in the TRP channels. We don't know. So.
Hi, thanks for your really interesting talk. I just wondered if you'd heard of Dr. Derek Lonsdale. He's written a lot about what you've talked about today, about the population of women who've been affected by HPV vaccines, and also noting the clinical similarities with ME. And he noted that one of the things that he thought was maybe a contributor was in the vaccine there's anti-thiamine. There's something in the vaccine that destroys thiamine, and that he's treated some of these people with high doses of thiamine and found that it's really helped. Maybe not a cure, but it's seen as a contributory factor, and a lot of the symptoms of POTS and orthostatic intolerance, fatigue, mitochondrial dysfunction, he said can be partly explained by this effect on thiamine. I just thought I'd mention
I don't know specifically about that, but I know that there are lots of different ideas out there on the internet, and there are very, very few control studies. And that's why I wanted to do control studies and get vaccinated girls of the same age, because other than that, I don't think that you can use it very much. So I would, as I go forward, I would do randomized control studies of everything that we think would have an effect.
All right, thank you very much.